Hello again. I'm Susan Amara, the president of AAAS and the chair of this year's annual meeting. I'd like to welcome you all to today's plenary session featuring Dr. Katalin Karikó. We're thrilled that Dr. Karikó can be with us to discuss her remarkable career and one of the most important scientific advances of our generation, one that is truly leading us to the other side of a global pandemic. Today's session is brought to us by the generous support of Johnson & Johnson, a member of the AAAS corporate circle and a longtime supporter of the AAAS annual meeting. J&J's support of some of AAAS's flagship programs, including our mass media fellowships, now in their 45th year, and tomorrow's Minority and Women in Science Award ceremony have provided really critical funding to our programs that powerfully advance our mission. Johnson & Johnson is represented by a longtime friend to AAAS, Seema Kumar, Global Head of the Office of Innovation, Global Health, and Scientific Engagement. Following Dr. Karikó's talk, Seema will lead us in a fireside chat. It is now my pleasure to hand things over to Seema.
Thank you, Susan, and hello, everyone. It is such a great pleasure to be here at this AAAS meeting. I wish we could have all met, in person, because I always enjoyed so much being in person at AAAS and doing all of the networking. Unfortunately, we have to do it virtually, but thanks to technology, we can. And so I want to, first of all, thank you for joining us virtually. I hope everybody's staying safe and well. Well, this plenary is going to be inspiring and energizing, to say the least. And it's also going to be a reminder that science is 1% inspiration and 99% perspiration. Perseverance is sometimes half the battle of science. And at the end, it's always worth it, as you will hear from Dr. Katalin Karikó. I'm so honored to be here speaking with and introducing Katalin Karikó, Dr. Katalin Karikó, who I met actually September of last year when we presented her, as well as her colleague, Dr. Drew Weissman, with the Johnson & Johnson Dr. Paul Janssen Award for Biomedical Research. The two of them were honored for their groundbreaking work on mRNA research that enabled the development of several COVID-19 vaccines. And of course, it also holds tremendous promise for future applications in both vaccines as well as therapeutics. The work of Dr. Karikó began several decades ago, and despite her belief in the power of mRNA, her research was not fully supported or funded. And flash forward to 2021 with more than 4.79 billion people across the globe having received at least one dose of the COVID-19 mRNA vaccine, the world could not be more grateful for her perseverance and for her belief in continuing to stay the course on mRNA research. We'll hear a little bit more about her story during our fireside chat, but in the meantime, I will first turn it over to Dr. Karikó for her presentation. Katalin, can I turn it over to you?
Yes, thank you. Thank you, Seema. So I am very delighted to be here. Actually, I am in Philadelphia, and I wish that all of the listeners and watch of this program would be here. But today I will present, and if I can have my slides here on the screen, then I can tell you about the development of mRNA for therapy.
Do you see it?
So I just put there a quote: "The path of scientific discovery is never straight line. It has twists and turns in every juncture. And path of scientific what I suggest to those who are at the beginning of their career to stay on the track, and no matter how long and winding that road might be." So can you make it that I can see my slide? Because I can just see Seema at present.
Oh. let's see.
I don't know. Okay. I can navigate from here. I have it. So, talking about the mRNA development for therapy. So the next slide shows that actually it started with the discovery of the messenger RNA, and this was in 1961, and it took 60 years that we had the first mRNA vaccine developed and approved by the FDA. And during this 60 years, the major important milestone were 1978, when isolated mRNA was delivered into mammalian cell and it could be translated. In 1984, when first time messenger RNA could be synthesized in a tube. In 1990, when the messenger RNA was injected into in the mice, and then the mRNA translated to the protein. 2000 was when we discovered that actually the mRNA was inflammatory, and 2005 we come up with a solution for that. And 2017, when LNP formulated modified RNA formulated in lipid nanoparticle was used as a vaccine. I mostly remember all of this. The first one, not in 61, I was just 6 years old and I couldn't even read in Hungarian, not that English, so I was not reading Nature and other journals, but the rest of it I remember because I was following this field for that long. And even before discovering the messenger RNA, the next slide shows that scientists wonder about that what could be that, how could it be that in the nuclei, the chromosome is present and then actually the translation occurring somewhere in the cytoplasm at the ribosome, and how the information is coming from the nuclei, from the chromosome to the ribosome. And so just making interesting experiments, and of course realizing that the ribosome has some RNA. And so what they did actually using the bacterium, the next slide shows what they have done is that they isolated DNA from bacteria, and then they also isolated RNA from the same bacteria. What was interesting is here showing that these different bacteria has different GC content. And when they looked at the RNA had the same GC content. So the scientists, actually this was Spirin and colleagues in Russia, they realized that this RNA cannot be, the messenger RNA, because it seemed like it was all composed the same. Today we know that it was all ribosomal RNA. So the scientists had to look further, and the next slide shows that they published in 2 papers. The next slide shows is that it was published in Nature. And what was interesting, you can see that both of the paper mentioned that it is an unstable molecule, because in that case, for example, in bacteria, it is a couple of minutes and the RNA degrades. So of course, scientists now that based on these discoveries, established what we know today. The next slide shows it that what is the information flow. So that the information is coming from the DNA. On the next slide, please. And it transcribed to the RNA, and RNA is translated to the protein. Of course, the scientists could, identify the RNA, but they could not synthesize it. So what they did, they could isolate, and that's what they started to work with. So the next slide shows that what they have done. So they realized that reticulocytes, which is the precursor of the red blood cells, enriched for one type of RNA, and they could separate this RNA. It separated, it was between the tRNA and the 18S RNA, and this fraction they collected, and actually this contains the beta globin, which is very enriched in the reticulocyte. And then they translated this in rabbit reticulocyte lysate, and they identified that it is mouse hemoglobin. They used radio labeled amino acid to detect that. And in a later studies in 71, and it was shown that when the same isolated RNA injected to the frog oocyte, it also translated to this protein. So these were the first RNA delivery experiments in very early 60s. While scientists, try to develop assays that how to deliver, other scientists, the next slide shows, were working on to figure out what could be the structure of this RNA, this new messenger RNA, which was discovered. And in 1975, probably was the year of the cap, because they discovered independently here in New Jersey, in Japan, and in Australia, that it has a 5' very interesting they called in this paper in the bizarre 5' terminus. And I put here the picture of Jenny Thomas, who is organic chemist, and he synthesized the cap structure and sent it to the scientists to identify, so they have a reference material. And Jenny Thomas became my PhD advisor. So when I started there in my PhD in 78, actually, I knew that the messenger RNA has a cap structure. Here I may mention that in the same year, Bernard Bonst at NIH also discovered the enzyme, identified that vaccinia virus has a specific protein, an enzyme, which can cap the viral RNA. Scientists advanced further, and the next slide shows with the delivery, because they were excited to deliver the RNA. So, many of you are surprised that the RNA was delivered, and which is something very new. But you can see here that 1978, they already have done that. In this state, at that time, also they used isolated isolated rabbit globin RNA, and this was from the rabbit reticulocyte lysate, rabbit reticulocyte. And what they did, actually, they put it into the liposome. The liposome was actually invented in the same time that the RNA was discovered. It was 1961. And Oleg Benham was the father of the liposome. And here the scientists put around the RNA, and this protected this liposome, which was phosphatidylserine ethanolamine containing phospholipid liposome. And they put, it helped the RNA to fuse to primary cells. Actually, they use here mouse spleen lymphocytes, and they could detect with immunoprecipitation that the RNA was translated. So this was published in two paper in Nature in 1978. So I consider that was the first RNA delivery experiment. We were reading about, actually, in this experiment, and was excited. And the next slide shows, now that my involvement is started here, because in 1970s, I started to work in the lipid laboratory first, and there we did generated liposomes, and we delivered at that time. It was plasmid. We did not know how to, or we didn't try to isolate RNA. But for me, I am mentioning this time because it was so important. This was the first experiment I was doing with mentors, and it influenced me. Later, I keep thinking about this very first experiment. And so I did a lot of lipid-related research, but when I finished my studies and I started my PhD, actually, I went to the RNA laboratories. And here, actually, not working with RNA, next slide shows, it was, I was working with a small 2', 5' linked oligoadenylate. And the next slide, you can see that. And these molecules was, I started in 78, and they were just discovered in 1977. Ian Kerr in London discovered that this short RNA, 2', 5' RNA, is responsible for the interferon-induced antiviral effect. And we decided that we very much need an antiviral compound. And, even today we know that how important antiviral compound is. So my role was to make this molecule enzymatically, chemically, and this oligoadenylate synthase was the enzyme which can, in our body, can polymerize from ATP. And I might mention here the oligoadenylate synthase is very important antiviral molecule. This is an enzyme in our body, and those who actually could synthesize less of this enzyme, they were more prone to get seriously sick from coronavirus, which turned out. So the function of this small molecule is that it activates RNase L, a latent nuclease, and then it degrades the RNA, all of them, not just the viral RNA, but that was what was believed at that time. So my love for RNA started here in the 70s, because I learned how to handle them, how to label them, and so it was an exciting time. I moved to the United States, and I still worked on the same 2' 5' link molecules, but when here the important antiviral compound would have been needed, that the HIV was very serious in 1985, 86. And but this molecule, this 2' 5' linked molecule, was not good for that. So when we, in the next slide shows that in this point, here in 86, we started to use double-stranded RNA. This was a mismatched double-stranded RNA, and we tried to induce the patient interferon system. Unfortunately, it did not have the HIV patient, because the interferon mechanism is compromised, and so we couldn't induce that. So, but by that time, it is about 88, when I am already working with short RNA, with the long double-stranded RNA, delivery problem, and others, and meanwhile, in the field, at the same time, started to move on, and that was the mRNA, because as I'm showing the next slide, in 1984, when mRNA could be synthesized in a tube. So it happened, as the next slide shows, in the laboratory in Harvard University. Douglas Melton and Paul Krieg performed this experiment and published in 1984. Interestingly, so what was important is that they realized that using phage RNA polymerase is the answer to make RNA in vitro. Prior to that, you can see publication where bacterial RNA polymerase was used, but those, problem was the promoter, the fidelity. Here, the phage polymerase required very short promoter. It has very high fidelity, and so that's what they used. That first RNA, what they synthesized and published, it was coding for human beta interferon. And they did the capping with the capping enzyme from Bernard Moss. And then, so the RNA had the characteristic 5' UTR, 3' UTR, poly-A tail, and the coding sequence. So when they synthesized, they injected, the next slide shows, into the frog oocyte. And when they cultured these, they injected a couple of frog oocyte, and when they cultured these frog oocyte, those secreted functional interferon beta, and they were functional, they inhibit viral proliferation. So this was a very important breakthrough, and not just because now that we could make RNA in a tube, as we designed, but it was also because this was a time when molecular biology advanced and became the tools. For example, here the SP6 RNA polymerase was commercially available in the same year when it was this was published. In addition, I might mention here that at that time, I was working at University of Pennsylvania, and I requested Paul Krieg for this plasmid, and he put in an envelope and he sent me. There were no MTA was signed at that time, so it was like the old days when everybody tried to have the others. The next important step in the next slide shows that instead of, injecting individually a cell, that of course is not feasible, but, the lipofectin was introduced. And it was 1987, again, when the publication came out, then the product was commercially available. And other products the same way, the Trizol, for example, Chomsky published it, how easy now to isolate RNA with it, and it was also available. And what was important that this lipofectin is a positively charged lipid, and nucleic acid is negatively charged, and it was mixed, protected, and then delivered to the mammalian cells. And the first time RNA was, next slide shows, was Robert Malone and colleagues delivered with RNA to the mammalian cells. So compared to the 78 publication where I demonstrated that, RNA was delivered in primary cells, here it was the RNA was not isolated, but rather it was in vitro transcribed. But otherwise, it was a very similar experiment. So next step was in our field was 1990, as the next slide shows, and the following slide, because that was when John Wolf published that he injected mice with luciferase-containing RNA, and it was a naked RNA because the lipofectin was not really used. Lipofectin helped move forward the project in cultured cells, but in animals, those were toxic. So most of the in vivo experiment was in naked RNA, just like here. And subsequently in the 90s, the next slide, I listed a couple of major findings in 92 when vasopressin mRNA was used in rats, and it was Floyd Bloom at Scrip could fix this special Brattleboro rats and treated them successfully. 93 and 94 was the two papers when first time messenger RNA was used as a vaccine for infectious disease, and that was for influenza. For Martinon and Maolian, they were working in France, and then actually they used liposome, similar what we were doing in Hungary. So and delivered messenger RNA. And subsequently, most of the studies were focused on cancer vaccine, and that was in 95 was the first time. First paper came out, Curiel, Conry, and Alabama. They used naked RNA for vaccination with cancer antigen. And Eli Gilboa at Duke University did a lot of studies, and he used human dendritic cells, and he generated the vaccine ex vivo, so that he moved on and established a company, Merix. Merix was in 1997 was the first RNA company, and they initiated clinical trial where they generated human dendritics from human PBMC dendritic cells and treated them ex vivo with tumor-specific messenger RNA, and those were injected back to the patient. And later, this Merix became Argos, the company, but unfortunately, their clinical trial were stopped due to futility. While all of these scientists were working on in vivo studies, I joined cardiology, and the next slide shows in University of Pennsylvania, and I also started to use messenger RNA together with Elliot Barnett, who is a cardiologist and actually working in Johnson & Johnson. But at that time we worked together at the University of Pennsylvania, and we were interested in intimal thickening, and that's what we generated messenger RNA coding for urokinase receptor. And I am just showing this example. We did other studies, but the urokinase receptor, to be functional, needed extensively modified, and so glycosylated GPI links, so that and mRNA was delivered to the cell, and then cell knew what to do with it, and then we could measure functional receptor on the cell surface. So at that point, we thought that, yes, the RNA will be good for something. But unfortunately, Elliot left, and so I moved over to neurosurgery. And the next slide shows that what we tried to do here, we did a nitric oxide encoding RNA, try to treat, for a goal was to treat a patient who had subarachnoid hemorrhage and get a major blood vessel constriction, and maybe local delivery of the nitric oxide synthase would help what we argue. So again, mRNA is not the drug. The mRNA codes for a protein, and usually that has the beneficial effect, could be therapeutic. But in that case, for example, it is an enzyme which codes for a product which has like the nitric oxide with a millisecond half-life. So that case, the nitric oxide is what is the drug. We also synthesized different kind of RNA because these are antioxidant enzymes encoding RNA, catalase, glutathione peroxidase, superoxide dismutase, because it was thought at that point that maybe the stroke patient would benefit if antioxidant enzymes would be higher level present. So we did a lot of experiments, and meantime we constantly improved the mRNA on a way that more protein was produced from it. So I was working on neurosurgery, and actually I worked there for 17 years, so all of the way to 2013. But meanwhile, when David left, and I met Drew Weissman, actually, and the next slide shows that I met Drew at really at the Xerox machine. And this was 1997, when we still copied papers because it was not available digitally. And if it would happen 2002, where all paper I downloaded digitally, so I wouldn't met him, but here he was, this new guy on the floor, and then we were started to talk, and then turned out that he wanted to make a HIV vaccine. I was working with RNA, and we thought that we can try work together and try whether mRNA would be a good vaccine. And so generating the gag mRNA and delivering to human dendritic cells, Drew was working with human dendritic cells. This was very new at that time. Ralph Steinman discovered, and so he generated from PBMCs, and when he delivered the mRNA I generated for this GAG-specific mRNA, he was very happy because a lot of protein was produced from the RNA, and all of this activation marker of these dendritic cells was up, and the inflammatory molecules were secreted by these cells. And of course, me, who was working in neurosurgery and wanted to use the RNA for therapeutic purposes, so coding for therapeutic protein, I was not happy that this RNA I am making inflammatory. And so we decided that we will figure out why it is, because we were in, even in 2000, when we published this paper, 2000 were not known, only just we knew that double-stranded RNA induced interferon, but even that time it was not known how, because it was 2001 when the first time toll-like receptor 3 was discovered, and understand that double-stranded RNA activate toll-like receptor 3, and this is how interferon is induced. So we try to figure out also that how could it be, the RNA which I am making, and I thought that the RNA I am making is the same which is inside all of our cells, is inflammatory. And then we argue that maybe because the RNA is coming from outside to the immune cells, maybe it senses as injury related or invader or whatnot. So we wonder that whether the RNA which is inside the cell, whether if we would take out and then now try to put into immune cells, whether it will be inflammatory. And this is what we did. And the next slide shows when we isolated, generated monocyte-derived dendritic cells and isolated different kind of RNA from the mammalian cells. And in purple, you can see that those are the in vitro transcribed RNA, which I was always doing in a simple in vitro assay, and the isolated RNA in blue, that those were not as inflammatory. We measured TNF-alpha as a marker for it. And the E. coli RNA was also quite, total RNA was quite inflammatory. And so we thought that maybe these RNA during the injury, released from the cell, they can instigate some inflammation. But what was more interesting for us that the tRNA, you can see that the tRNA was not inflammatory at all. And in the next slide, you can see that the tRNA, and we knew that, has a lot of modification, nucleoside modification. So this is the thing which gave us the idea that could it be that nucleoside modification could suppress the immunogenicity of the RNA? And this is, we try to answer that. And of course, how we could do that, how we could make our mRNA and incorporate a modification, and which kind of modification we should induce. And the next slide, just to scare you a little bit, I show here what kind of modification there in an mRNA, and there are more than 100 different ones. And these are incorporated after the RNA is made from the 4 basic nucleotides synthesized in our body. Different enzymes, they change and modify, and those enzymes were not known. We just couldn't order to put somehow to our RNA and see that whether it will be less immunogenic. And so we didn't know how we should proceed, and came on the idea that we might have to order nucleoside modified or triphosphate, the modified nucleoside, which is triphosphate one, and try to incorporate to the RNA. So that was we have done. And the next slide shows that what was commercially available, those 10 different nucleoside triphosphate. And why we ordered this one only? Because we insisted that we are just working with modified nucleosides, which is normally present in our body. And the reason for that, because I remember from the early 90s, 93, when it was published that fluorinated uridine containing, this is called fluoridine nucleoside, when they delivered to human from a clinical trial, 5 patients out of the 15 volunteers died. And they did not understand for more than 10 years that why it happened, because this molecule was not toxic, not to the mice, not to the monkeys, not even monkeys. So it took, in 2006, when finally it was realized that there is a special nucleoside transporter which is only present in the human, and this middle of the coding sequence has a mitochondrial localization signal. And what happened is that all of these unnatural modified nucleosides were pumped into the mitochondria, and the patient died from acidosis. So we don't want to do any kind of unnatural one. We want to use only these 10 different nucleoside triphosphate to make RNA with it, because it is present in our body, and a human body would know how to handle. Of course, we didn't know that whether it can incorporate by the enzymes. So that's what we tried in the next slide shows, when we were successfully could make at least 5 of them to incorporate to the RNA. And the next exciting step was to find out whether these RNA now that containing these different modified nucleosides, whether they will be less immunogenic. Of course, we were excited because we didn't know at that time, whether we need all of the modification which was in the tRNA or one of them. We didn't know what we would need. And the next slide shows that we were very happy that finding that some of these nucleoside modified containing RNA were not immunogenic. These were now measured in isolated primary cells. These are isolated primary dendritic cells, so it was not generated, so it was isolated from the blood. And then we found that those which had modified uridine, those were not immunogenic. And so we wonder that maybe uridine is somehow nature selected, that detecting and the immune cells detecting to as a foreign material. But when it is pseudouridine, 5-methyluridine, 2-thiouridine, or whatever was the uridine was modified, it was not immunogenic. Of course, I wanted to use the RNA for coding for therapeutic protein. Drew wanted to use for vaccine, but both cases we needed that this RNA should be translated. So the next slide shows that luckily we found that some of the nucleoside modified uridine containing RNA was translatable. What was surprising is that the pseudouridine containing RNA translated so efficiently. 10 times more protein was produced than from the conventional, which shows in the blue bar there. But the pseudouridine translated very well in primary cells and all. And whereas the 2-thiouridine was not translated. So subsequently, now that we focused all of our study to the pseudouridine containing RNA. But when we did some experiment, we noticed that we have some contamination present. So the next slide shows that when using the double-stranded RNA-specific antibody, we could detect in a dot plot assay that double-stranded RNA was present in all of our RNA transcript. And so it took about 2 years and a lot of work to figure out how to get rid of that double-stranded RNA, which finally we could manage in 2011. And we established an HPLC purification procedure, and we could purify RNA. It was very important because in the light blue, the blue bars, you can see that how much translation was improved when the RNA was purified, modified. So finally, we could get a very high quality RNA, which was nucleoside modified, which was purified, and we moved on. And the next slide shows that we want to see that whether we can, this RNA can be translated in vivo. At that point, we used, in the next slide shows that we used transit to deliver in vivo to the animals. In that time, we didn't have the lipid nanoparticles. We just had this transit, which is commercially available. And what you can see in the low panel is that when we use pseudouridine containing RNA, and very small amount we injected to the animals, 0.1 microgram mRNA, for a couple of days we could measure erythropoietin actually in the blood. When we did experiment and we just injected 0.1 microgram messenger RNA coding for erythropoietin, which is responsible for red blood cell differentiation, 30 minutes after injection, we already could measure the EPOs present in the circulation. So the RNA is taken up by the cells instantly, translates instantly. The speed of translation is like 5 amino acids per second. So depending on the length of the protein, you can detect very, very quickly. You can see on the blue line there that when we use the conventional uridine containing EPO mRNA, that not translated as efficiently. And these were all HPLC purified mRNA molecules. On the right panel, you can see that when we injected this pseudouridine containing RNA, the hematocrit of animals increased, and the weekly injection, the very right panel shows that the weekly injection of erythropoietin coding RNA, small amount, could maintain hematocrit at high levels. So the RNA we injected, it was functional. I have to emphasize, of course, we didn't inject it in the kidney, we injected it intravenously. We also did some studies where intramuscularly or sub-Q we injected, and it didn't matter where we injected, we will always could measure EPO circulating in the blood and also the hematocrit increase. The very important panel is in the middle one to show that even in animals, the nucleoside modified RNA was not immunogenic, whereas the uridine containing RNA was. This was 2012, and in that time, you know, it seemed that I have to move on, and I moved to BioNTech to Germany. And next slide shows that although the new building was not there yet, we were on the campus and we were a very small company. And I moved there because I wanted to make messenger RNA coding for therapeutic protein, and Uğur Zahin established this company, and he already, in 2012, already messenger RNA entered in clinical trial as a cancer vaccine. And I thought that if I want in my lifetime to see nucleoside modified RNA as used as a therapeutic, I have to go to Germany and try to do here. Here and just showing one of the example when I was there, and I'm still there actually, at a company that we started to make bispecific antibodies. And these are very short half-life this protein has. So the advantage of the mRNA in this case, not only that it is very cheaply, the animal can translate the RNA and generate this bispecific antibody, but also that this short half-life, that due to the short half-life, very frequently had to inject here in the animal, but in human already the protein, the bispecific antibody is human use, and those patients need pump put to the arm, and then even the pump had to be filled up every second day with this protein. So it seemed that if they would have the RNA, they would not need a pump, and then they do not need such a frequent injection. So here we show that it is very effective, and it was the bispecific antibody recognized partially the Claudin-6 tumor antigen and CD3, so that it put close proximity the T cells and the tumor cells. And we have done a lot of study here. Actually, we moved there with my colleague Hiromi Muramatsu from Penn, and we did a lot of study. And at the 2 years, we were just doing the same thing we did at University of Pennsylvania, but now we had to do it in a scalable manner. And so that was, one of our focus there, so that the purification, like the HPLC purification, couldn't be scaled up. So we had to come up with other type of purification. We also had to, the capping of the messenger RNA, which is very critical to have a cap1 here also shown. Enzymatically, we put there, but it was very tedious, so that we had to come up more improvements. So the next slide shows what we were doing at that time, doing a lot of optimization. And the 3' UTR, 5' UTR, the coding sequence, so we did, when we did all kind of optimization, purification, we could, as the panel shows, we could have from the mRNA made more protein, longer time, higher level. On the next slide, I will just show you that what some codon optimization could result. So that we used pseudouridine in our experiments, so that when the inflammatory uridine are there, with showing in blue here that the pseudouridine containing RNA would not be immunogenic, but and some used instead of using pseudouridine, rather than eliminate the uridine from the messenger RNA. And however, and that making the RNA GC rich, but one problem with this one, that you cannot 100% eliminate uridine from the RNA, because there are amino acid, and those here that half of the amino acid needs uridine as to code. And some, the phenylalanine need minimum of 2 uridine. So in every time, if you are doing codon optimization with this way, the RNA immunogenicity depends on what is the composition of the protein you try to encode. And I also mentioned here 2 reasons why I wouldn't codon optimize actually for infectious disease related vaccines, because you might lose with this optimization out of frame cryptic epitope, which is very valuable, or you are generating a new epitope, which is useless. But so that's optimization, codon optimization had to be handled very gently. While we were working actually in in Germany, my colleague here in Norbert Paddy, the next slide shows, he was proceeding with formulating modified RNA to the lipid nanoparticles. And he published this paper together with Drew Weissman, and I was also included. And this publication where messenger RNA encoding the Zika virus specific proteins were done, and this was one method of pseudouridine containing RNA, just like in the COVID-19 vaccine. The lipid nanoparticle was very similar, made by Aquitas, were formulated, and these were tested out in mice, and it was also in monkeys, and it was very, very effective. And single injection of this mRNA, this LNP mRNA, protected the mice. And very small dose, 50 microgram, was also sufficient to protect the monkeys when it was challenged with the Zika virus. And I might mention that today the BBVA award, which given out by Portugal, and for the best paper of the year, for the looking at 10 years, what was those papers which had the most impact. And today this paper get the award for 2021 from the BBVA award, because the importance that was the first paper when lipid nanoparticle formulated nucleoside modified mRNA was demonstrated that how powerful vaccine it could be. So from we went on with Norbert and published and performed more experiments. This was for Zika virus. It was also later with influenza and HIV. And we also demonstrated and had proof why nucleoside modified RNA was critical. And as recently as this year, we also published why was the lipid nanoparticle is critical. It is not just wrapping up the RNA, helping to protect, helping to deliver to the cell, entering to the endosome in the immune cells and helping to release it, but it is also containing it is an adjuvant. The ionizable lipid act as an adjuvant because the mRNA, messenger RNA, is not immunogenic. And why it is important, because the messenger RNA would induce, if it is conventional RNA, it would induce interferon. And if the interferon induced, as we demonstrated, follicular T helper cells will be not present. And then result that there will be no good antibody response. Maybe good cellular response, cellular immune response can be generated, but not antibody response. So this was the importance of LNP, the nucleoside modified RNA. And as the next slide shows, and I don't have to tell you more about this because we were sitting in the front of television and we knew when it was, the sequence was became available for the SARS-CoV-2. We watched what happened when all of these companies started to, 150 companies at the same time started to make a vaccine, and how the phase 1, the phase 1, 2, and phase 3 trial ended. And so as a result of it, we know that within a year, we already could have a vaccine, which is very safe and very effective. And last year, August was when it was fully approved, the BioNTech Pfizer vaccine. And I might mention that it is a public domain that BioNTech started to work with Pfizer in 2018 to develop an influenza vaccine. So when people learn about, the development of messenger RNA for vaccine, they thought that this is the first time, that compound, this messenger RNA, is used. And as I told you already, many studies were done, but many of you will not know. And the next slide shows that actually the nucleoside modified mRNA was already in a clinical trial. The first patient actually was injected in February 5, 2018 with the messenger RNA coding for VEGF-A. And this study started actually from Harvard and University when Lior Zhangyi and Kenneth Chen, injected VEGF-A mRNA to the failing heart of mice, and they demonstrated the beneficial effect. And AstraZeneca and Moderna proceeded with this finding, and STUD clinical trial was already underway, one of them using for wound healing, for necrotic wound of the diabetic patient, and the other was the heart failure treatment, which started prior to the COVID-19 vaccine. And the next slide shows that this was the most advanced, mRNA therapy, because it was already in a phase 2 trial. And this year, January, we learned that the patient who participated in this trial, and this was a elective coronary artery bypass surgery, where during the surgery they injected 30 times naked RNA coding for VEGF-A to the heart. And this on the slide on with marker on the right, you can see those labeling. And they injected the RNA, and in the link there shown there is you can hear the patient who are talking about their experience, that how their life was improved and their heart was performed so much better. And so now it is in this clinical trial advancing to phase 3. So there is a success already in other field of this nucleoside modified RNA. This was also a 1-method pseudouridine containing RNA. And another field, not this was for the heart. Another was the next slide shows that was for the liver. And in this case, if the next slide please. In this case, Cas9 mRNA treatment was used by the Intellia company to treat this patient who had amyloidosis, transthyretin amyloidosis. And this is a very serious disease. And there were already the Alnylam, actually, when they developed siRNA, that was the first what they targeted to treat this patient. And what happened is that they have a malfunctioning protein and coded in the liver, and this protein, when it is made, it is aggregate, and then interrupting this production of this toxic protein would be very critical. And of course, Alnylam targeted the messenger RNA, but here the Intellia used the Cas9 RNA, and the next slide shows that this mRNA, what they delivered, they contains the mRNA codes for the Cas9 protein. The next slide, please. And then a guide RNA. When it delivers, inside the cell, the Cas9 RNA translates to the protein, and complexing with the guide RNA finds the critical target in the liver cells, the hepatocytes. And maybe the next slide shows that, how it interrupts, and eventually this toxic protein is not produced. And the result of this trial, which was published last year, is shown that the patient in the blood, the level of this very toxic protein was dropped. And so here already 3 patients were permanently treated and cured. So that's another big success of this nucleoside modified RNA use. I mentioned here one more example because the next slide shows when intratumoral injection of mRNA was used for cancer treatment. This is not a vaccine because the messenger RNA is not coding for cancer antigen, but rather it was cytokines, a mixture of cytokines. We performed this experiment, and here is shown only in animal studies, on the mRNA coding for IL-12, interferon alpha, GM-CSF, and IL-15 sushi. And this messenger RNA, when injected to the cell surface closed tumor, like a melanoma in an animal setting, then not only so what happened is that the tumor, cell surface closed tumor, which is cold, became hot because these cytokines locally were secreted, immune cells were rushed there, and then when they were in the circulation, then they could eliminate distantly located tumors. Here in the picture shows on the right that it eliminated the tumors from the lung. This is just the animal studies are shown. This is with Sanofi. It is advanced to human trials, and hopefully the patient will be cured by that. So we have to wait for the result. And so then finally, I want just to mention to you that in the last slide shows what is now is 2022 and what is beyond. So I mentioned all of these clinical trials which were successful. In addition to that, there are other trials are ongoing when messenger RNA codes for monoclonal antibodies. We already heard, in the news, we can hear that for HIV, for flu, already clinical trial using nucleoside modified RNA, LNP, is already initiated and first patient already injected. And more genetic disease are targeted with using enzyme which can edit the genome. The preclinical trial ongoing, and those are, and let's just try to mention here, even 4 years ago, we already on the messenger RNA therapy meeting, we already heard about using messenger RNA to vaccinate against malaria. Herpes simplex, I showed the data for Zika virus. Last year, we demonstrated that using multiple sclerosis specific autoantigens, we could induce tolerization in 2 different multiple sclerosis model animals. And so this is also a new field. We published how VEGF-C can help formation of new lymphoid vessel and fight edema. And more papers, like just last week was one about bone healing using a BMP-specific mRNA. So this is a platform. The messenger RNA therapy this is a platform. Many diseases can be treated. There are, luckily, more attention was paid now. It is the advantages that it doesn't matter, it is a vaccine or any kind of RNA, you can use the same settings, only the order of the nucleotides, which is incorporated based on the template, is different, but otherwise the messenger RNA is the same. Of course, the formulation is important and it is different because for a vaccine, when we are using a formulation lipid nanoparticle, those are containing the adjuvant, and maybe it is not useful for all. So for other formulation, for other treatment, we have to use other formulation. For example, for tolerization, where we didn't want to activate the immune system. So lot of effort is put on to make the formulation targeted to selected cell groups or organs, and this is the future to make it more specific, the therapy. But the other advantage of this mRNA therapy is that it is affordable because of the human or the patient will make the drug translate, which is the protein-based therapies are very expensive because purification of the protein is very tedious to figure out what is, even if it is protein is the final drug, for screening, the messenger RNA can accelerate tremendously the screening time because, and put through. So many things can be done, and we will see the future that what it holds. But now that more attention on the field and more money is coming, so hopefully a year from today we can talk about more successful clinical treatment. And with that, the next slide just shows all of my colleagues, and I want to thank them all for helping me on this journey and those long winding road that I stayed. And thank you very much for your attention.
Thank you, Katalin. That was just such a thorough and extraordinary talk. You took us along on this whole journey throughout your own career and finally ending up at where we are today. So thank you for that. What I wanted to ask you actually is, since we've covered your science in great detail, I wanted to ask you questions about, the career in science and women in science and things of that nature. So let me start by asking you, first of all, you left Hungary in 1985 and you moved to the United States and continued to pursue your research. What lessons would you share with others who want to immigrate to the US and continue to pursue a career in science? And how does your experience compare to those who are trying to come to the United States and work in science today?
'85 was a long time ago. And of course, a lot of things changed. And so for me, when I think back that how important was for me, like being at the University of Pennsylvania, the number one importance for me was to listen to those lectures, that the best scientists came, and I was just amazed. And every week I had to limit myself to listen not more than 2 lectures, because otherwise I have to work in the lab. So today you can be in your home country and you can listen to all of these lectures, and you can have access to all of these publications and others. So that's also a very important part. And as the process that you are coming to a country and then all of a sudden everything is new, you have to adopt so quickly. You learn these things. And what is science? Every time we have a new problem, now we have to purify how we would make it. And then, it is a good exercise to learn that how to quickly adopt and how quickly you can come up with solution for anything. First in life, because you have to adopt a new home, new environment, and then it is similarly in the laboratory, that when you are doing different things, and it is much. So what happened is a lot of people like to, it is very comfy corner, and they don't want to leave, and they don't want to, and then it, if they leave, then that's immediately. Have a lot of experience. I personally never wanted to come here, but no, I don't mind, obviously. But,
so you're back and forth always now.
Yes, but so it is. I like the open-minded, here in the United States, for example, that, but maybe, it changed also in other corners of the world. Oh. But, that everybody was so helpful. As I mentioned during the presentation, that the people who I never met, they just sent in an envelope. We received the plasmid cut out and soaked in, the little bottom paper, because they would just want to help. And so it was very uplifting.
Yeah, that speaks to an important topic that I know is close and dear to AAAS's heart, and being discussed all around the world, and that is the importance of international collaboration in the scientific enterprise. What are your thoughts on that? Because especially during the pandemic, we saw a little bit of a rise in nationalistic sentiments, but really science flourishes when there's international collaborations, and cross-country, cross-border science is very important, right? Your thoughts on that would be so welcome.
Yes, so this pandemic, and I put science in the spotlight, and then realization that collaboration is a must. And so that we can see already even every level, not just bench scientists who try to help each other, but we could see that even the large companies, if the other company had a lipid expert and reached out, to how to scale up, for example, the lipids, and so it was everybody's input. And if you see that our fellow Chinese scientists who sequence and then they distribute the data and how it helping everybody to work together, and for me also moving in Germany, actually, that in my team is very international. BioNTech is very international, like 57 nations was working there, and women and men, I have more women worked there, and so it is, and then those reaching out for more in the home country. I also try now that I have more time, to reach out back to Hungary, and maybe it will be very effective.
Well, yes, that is great, and I think that this is what we've always seen, and it continues to see the international collaboration. You brought up a really great topic that's very close to my heart, and that is the topic of women in science. So you are one of those women in science who has just made it to a place where people recognize you. Because a lot of times when you ask somebody to name a woman scientist, a famous woman scientist, people may not always think of it. What do you think we must continue to do to encourage more women and girls into careers in science?
Yes, so I think that, the children are curious, and then and boys and girls equally curious, and in school, they are advancing, and I don't know where the girls will fall out from this process, but one obviously, of course, when they, give birth, and it is hard for them, to handle all of the job they need, and so for me it was very helpful that we had affordable childcare, quality and affordable. That's what one would be very important to have, and I don't know that I try to encourage also children and girls and boys, it doesn't matter, that to study, science, because they have to realize what is this school is for, that to realize that what do you like, what you would like to do in the rest of your life, and that they have to find that, and that's what the school for, they do sports, do that, and they realize that what they like. Mm-hmm. And so that, of course, if they like this spotlight, which I am, probably they want to be an actress or some performing art, but if they like to solve problems and having fun with this and coming up every time to figuring out something, and having fun, then they should be a scientist. And so that's what I recommend them. And I don't know if they want to be rich. I don't know what they want, what they should, but what they would do, the money, it is more having fun is, I think, is more valuable. And having a goal to that your research at the end helps somebody is the, as ultimate reward.
Yeah, that is great. That, that it's because at the end of the day, science is fulfilling when it reaches society and solves a problem. And you must be so thrilled to have, something that you worked on almost your entire life to actually have had such a huge impact on the world. What do you think the future is for biomedical research, both here in the United States and across the world?
So I am very optimistic because, I can see who are around us, that, very bright young people, and but we need more because there are so many things to discover. And so I hope that, I know that I was not funded, but I can understand also that, it is I was, I don't blame anybody for that. I just blame myself. I have not done enough preliminary data or not explained well enough and whatnot. So I hope that more money will be spent, and because we realize, that here is a virus, we cannot see it. You need a certain assay to detect, and then it can stop our life on the way we knew it. And so that we, not just because of this, but that is very important to invest into science. And there are so many diseases could be cured if we would put more emphasis on it. And I know that the basic science is important, but the applied science, and I was a basic scientist, but I was always thinking what it would be good for, so that who is performing the experiment, maybe that's the person who can think the application most likely.
That is excellent, because you're right. I mean, we have not yet scratched the surface of all of the healthcare challenges we need to solve. There are so many diseases that still need, therapeutics and vaccines and cures. I have one last question, because one of the things that we've all talked about is, despite this great, wonderful thing about vaccines, and the vaccines are available for people, there is still hesitancy, and there is still a little bit of hesitancy in believing the science. What do you think we as a society, especially as a community of scientists, can be doing to increase and enhance trust in science?
Yes, so I realize that we have not done a good job in the past because we did not educate the public. We didn't talk about what the molecular biology is doing. Where our science today? Maybe it was papers or something, or articles about, but not a simple language. And maybe those who are making films, they were not doing about science. So that we have to do that, and we have to explain some simple way to the people that what is the science, what is the vaccine, how it acts, and one thing is also that to tell them that what we know, this we know, and that we don't know yet, and this is a process. This is and then the honesty, which we always, as a scientist, we are always honest, but maybe it didn't come through to the people, and then those people who had more time, who were just hopping from studio to studio and telling stupid thing, and anti-vaxxer, because they didn't have to go to the laboratory to work, and so they spread things which was absolutely not true.
Yeah, you bring up a very good point about, in the old days of molecular biology, we probably could have done a better job of explaining what is molecular biology and how it works. That's a great point. Your hope for the future, what is your biggest hope for the future? And do you think that mRNAs will turn into therapeutics, and are you working on that?
Yes, of course, I believe that the RNA will be help for a lot of treatment. Of course, it will be not good for everything. The RNA, no matter how you wrap it up, it won't go through several layers of cells. But it can help a lot of treatment of a lot of diseases, and I am very optimistic. I was always very optimistic, and believe that when I can see science and it really is not a belief, because we are scientists and we know the data, and we look at the data, and then we can conclude things. So, but I am very optimistic, but we have to work also to inspire the new generation, and explaining to them that what we are doing. And so being in the spotlight right now, that's what I try to do, when I go out to different countries, to school children, and to tell them about, the excitement, and what as a scientist we can experience. And I encourage them to find what they like to do, because, you know, if they like to do something else, then that's what they should select. Yeah.
That is just fantastic advice. And I'm so glad that you're using your time in the spotlight to continue to inspire the next generation into careers in science. And I know that everybody watching today and the entire AAAS community and the entire Johnson & Johnson community will be very happy, because we're all about inspiring the next generation of science, as you know. And I also wanted to let the audience know that even though we are out of time, I hope that you enjoyed, you know, Katalin's just phenomenally thorough, very good presentation, and also the conversation we just now had. If you wanted to listen more to the conversation, then please go to the J&J YouTube channel, where Katalin and I actually had a much longer conversation with each other in September when she won the Paul Janssen Award. And so for that information, please go to the J&J YouTube channel. And for those of you who are here at AAAS, in case you were not able to watch this, the replays are going to be available. And so thank you very much to the AAAS community.